Good morning. I hope you had a wonderful spring break and are ready to learn. If you have not completed the video and the questions on the video, please do so now. That will be a grade. If you have not completed the packet with the different ways to control microbes, please do that now. It will be much easier for you to get through the lecture if you understand the difference between disinfection, sterilization, sanitation, and aseptics. Please grab a learning target packet before you start the video also. If you want to use OneNote to take your notes, that's also fine. Just make sure you have those learning targets ready. I will try to point out as I go along what the learning targets are. You may stop and pause to take notes at any point, so I'm not going to pause much in this video. So, controlling microorganisms. The methods of microbial control used outside of the body are designed to result in 4 possible outcomes: sterilization, disinfection, decontamination, or asepsis. Sterilization and disinfection are used on hard surfaces or inanimate objects. You will disinfect your area before and after a lab using the 70% alcohol or a 10% bleach solution. Antisepsis is when you wash your hands or use hand sanitizer. Antisepsis is for tissue. They're not as effective, but they are designed to not be harmful to human tissue also. This should look very familiar. This lecture is going to be about the differences between the physical, chemical agents and the mechanical removal methods. I am going to go into more detail about each of these. Like I said, please make sure you know the difference between disinfection, sterilization, antisepsis, and decontamination. So, relative resistance of microbial forms. Primary target of microbial control are microorganisms capable of causing infection or spoilage in the environment or on the human body. Basically anything that can cause harm to us or to our food. The goal of sterilization is to kill the bacterial endospores. If you will remember when we talked about the components of a bacteria, bacterial endospores are very hardy, they're very difficult to kill, and because of this, they are the most dangerous bacteria that we run into. So the goal of any sterilization process is the destruction of bacterial endospores. Any process that kills an endospore will invariably kill all less resistant microbial forms. Your very first learning target is identifying the microorganisms that are most resistant and least resistant to control measures. So least resistant are enveloped viruses. This means they are the easiest to kill. Most resistant are the prions and the bacterial endospores. This means they're the most difficult to kill. Prions are proteins that affect the nervous system or the neurological system in an animal. And you may be familiar with mad cow disease. Mad cow disease is dangerous because of the prions. Prions are so dangerous that if something becomes infected, like surgical tools, they will decontaminate or try to sterilize those surgical tools, and then they will dispose of them. They will not try to use them again because they really just aren't sure if they've been able to kill them. That is how dangerous and how hard to kill prions are. Really quick, let's look at comparing endospores and vegetative forms. Endospores, obviously, they need a higher heat, a higher amount of x-rays, a higher amount of ethylene gas, and a longer time with the sporocidal liquid to die. Vegetative forms are much easier to kill. Lower temperature, 10 minutes with a glutaraldehyde. That's pretty simple. Agents versus processes. Processes are sterilization and disinfection. Agents are what we use in the processes of sterilization and disinfection. Bactericides are chemicals that destroy bacteria except for those that are in an endospore stage. Fungicides kill fungal spores. Virucides inactivate viruses. Remember, viruses are either considered active or inactive. So we are trying to inactivate the viruses. That's what a virucide does. And a sporicide is capable of destroying an endospore. So bactericide kills bacteria, where a sporicide destroys the bacterial endospore. Germicide or microbiocide is an agent that kills microorganisms, a very general term. This is your second learning target, by the way. Sepsis, the growth of microorganisms in the blood and other tissues, and asepsis is trying to prevent the growth of microorganisms in the blood or other tissues. Aseptic technique is a practice in healthcare that range from sterile methods to antisepsis. You will note that we use aseptic technique when we're doing labs, where you decontaminate or sterilize your work area and you wash your hands before and after we pull out the bacteria. Aseptics. These are chemical agents applied directly to exposed body surfaces, wounds, and surgical incisions to prevent vegetative pathogens. Antiseptics have to be effective and not harmful to tissue. Stasis and static, this is also part of your learning targets, means to stand still. Bacteriostatic are chemical agents that prevent the growth of bacteria on tissues or on objects in the environment. Fungiostatic is chemicals that inhibit fungal growth. Antiseptics and drugs often have microbistatic effects because microbicidal compounds can be toxic to human cells. So, microstatic is something that prevents growth. That doesn't kill it, but it prevents the microorganism from continuing to grow. And microbicidal means to kill or destroy. And remember that even a cidal agent doesn't necessarily result in sterilization. If you don't use it correctly, it's not going to sterilize. So what is microbial death? Death is the permanent termination of organisms' vital processes. In the video, it said that it was not necessarily trying to kill the bacteria, but to prevent it from reproducing. So if you have a bacteria that can no longer reproduce, one little bacteria that cannot reproduce is not going to cause you any harm. Modes of action of antimicrobial agents. Antimicrobials have a range of cellular targets. The least selective agents tend to be effective against the widest range of microbes. So heat and radiation, they can kill a wide array of things, but not necessarily as effectively as a selective agent that targets a very specific cellular component. That is going to be very specific to the target that it's working on. The name, the 4 categories of cellular targets for physical and chemical agents is one of your learning targets. The cell wall, the cell membrane, the cellular synthetic processes, and proteins are the 4 categories. Let's take a little bit deeper look at the different categories. So cell walls, they use chemicals to damage the cell wall. Basically, you're blocking what can go in and out. You're disrupting synthesis. You're disrupting digestion. Detergents and alcohol will do that. The cell wall is made up of lipids, and they're very susceptible to different detergents. Cytoplasmic membrane also has a lot of lipids in it. Detergents will break it up. This is disrupting things that can enter and exit the cell. The cellular synthesis are agents that can interrupt the synthesis of proteins. Basically, they inhibit the ribosomes from building new proteins, and if your proteins can't do enzymatic activities or other processes that are important to the cell, that's going to be very hard on the cell. Radiation, formaldehyde, ethylene oxide gas, these are examples of cellular synthesis. And the proteins, you're trying to denature proteins. Remember, if a protein is very sensitive to different pHs, to heat, and if you denature it, it's no longer active. Methods of physical control and heat. So the 6 methods of physical control of microorganisms is heat, radiation, desiccation, gases, liquids, and filtration. Gases and liquids fall under the chemical, but they are put into their own categories because they're very different methods. So methods of physical control of heat, you have moist heat and dry heat. If you have an elevated temperature, you are going to be microbiocidal, which means you're going to be killing your microorganisms. If you have a lower temperature, you're going to be microbiostatic, which means you are just enabling it from reproducing. Moist heat is hot water, boiling water, steam. Examples of this are autoclave and pasteurization. Dry heat are hot air or an open flame, which ranges from 160 degrees Celsius to thousands of degrees Celsius. Examples of this would be when we flame the loops, when we're trying to streak for isolation on a bacterial plate. Modes of action or methods of moist heat control. This is one of your learning targets. It operates at a lower temperature and shorter exposure times to achieve the same effectiveness as dry heat. Microbiocidal effects is the coagulation denaturing of proteins. Once again, examples of this are an autoclave or pasteurization. Methods of dry heat is they are trying to remove water from the cell, which will disrupt the metabolic processes and also denature the proteins. At very high temperature, it oxidizes cells, which basically burns them to ashes. Examples of this would be burning something to kill everything, and flaming your loop when we're using sterile technique during bacterial labs. So heat resistance and thermal death, endospores and vegetative cells. Bacterial endospores, like I've said, are very hardy. They're hard to kill. They exhibit the greatest resistance. Their destruction of the spore usually requires temperatures above boiling, and their resistance varies. So you're going to have a gram-negative bacillus or a clostridium, where they're going to have different resistance to different types of control methods. Vegetative cells are wimpier. They're not as sensitive to heat, and they can be killed in a matter of a couple minutes, where an endospore you may have to take up to 45 minutes with pressure and steam to get them to die. Thermal death measurements. This is one of your learning targets. Thermal death time is the shortest length of time required to kill a test microbes at a specific temperature. So basically, you're looking at a temperature, you get a specific temperature, and you want to know how long it takes to kill at that temperature. Thermal death point is the lowest temperature required to kill all microbes in a sample of 10 minutes. So now we're looking at time. You got 10 minutes. What's the lowest temperature we can use in this 10 minutes to kill our microorganism? Heat treatment of perishable substances must render the product free of agents or spoilage or disease without affecting the speed and cost of processing. So you're using the thermal death time and the thermal death point. Basically, when you're packaging specifically like meats and other fresh foods, you're trying to lower, you're not necessarily trying to kill the microorganisms, but you're trying to inhibit them from growing. This is why if you leave something out on the counter at room temperature, it will spoil much faster than if it was in the refrigerator.